this lecture, we are going to talk about part 2 of translation, and we focus on initiation. Main points of the lesson are prokaryotic initiation, eukaryotic initiation, and difference between prokaryotic and eukaryotic initiation. In the previous video, we discussed about prokaryotic and eukaryotic ribosomes. Ribosomes have double function, that is decoding function and an enzymatic function. The intact ribosome has 4 binding sites and polysomes. In this lecture, we focus on initiation. The process of protein synthesis has 3 distinct stages, that is initiation, elongation, and termination. Initiation of protein synthesis is essentially the same process in all organisms. Here we compare the bacterial and eukaryotic initiation. Translation in bacteria begins with the formation of initiation complex. Initiation complex that includes the small ribosomal subunit, messenger RNA, the initiator tRNA molecule charged with chemically modified methionine, that is called N-formal methionine, and initiation factors. Initiation factors are involved in initiation only, and they are not part of the ribosomes. In prokaryotes, the small ribosomal subunit is positioned correctly on messenger RNA due to conserved sequence in the 5' end of mRNA molecule, and this conserved sequence is called ribosome-binding sequence, abbreviated as RBS. And this ribosome-binding sequence is complementary to ribosomal RNA present in small ribosomal subunit. Then 50S subunit, it binds, forming an intact ribosome, and translation can begin. With the formation of complete ribosome, initiator tRNA is bound to the P site with A site is empty. Translation in bacteria is divided into 3 steps. Step number 1 is the small ribosomal subunit with initiation factors. It binds to the messenger RNA at AUG start codon. Now, how the small ribosomal subunit identify the AUG codon sequence? The leader upstream sequence of AUG codon is called ribosome-binding sequence, and this ribosome-binding sequence is complementary to ribosomal RNA present in small ribosomal subunit. It helps the small ribosomal subunit to identify the AUG sequence. Step number 2 is initiator tRNA binds to the start codon, that is AUG, and one of the initiation factor is released. GTP is an energy rich molecule, and GTP is used in protein synthesis. So, during initiation of translation, GTP is associated with initiation factors, and hydrolysis of GTP provides energy for initiating ribosomal complex. Step number 3 is when large ribosomal subunit binds to small ribosomal subunit to form an intact ribosome, the remaining initiation factors, they are released. So, the translation initiation complex is complete in prokaryotes. Now, initiation in eukaryotes, that involves 4 steps. Step 1 is the formation of pre-initiation complex. Pre-initiation complex, it includes small ribosomal subunit, methionine tRNA, initiation factors, and GTP. Step number 2 is this pre-initiation complex, it binds to a protein complex, and this protein complex is associated with 5' methyl guanosine cap of messenger RNA, and this protein complex, it scans the leader sequence base by base to locate the start codon AUG. Step number 3 is when the initiator methionine tRNA anticodon, it binds to the start codon AUG, it triggers the release of initiation factors. And step number 4 is hydrolysis of GTP that provides energy for the binding of large ribosomal subunit with small ribosomal subunit to form an intact ribosome, completing the assembly of translation initiation complex in eukaryotes. So, during protein synthesis, small ribosomal subunit and large ribosomal subunit reassociate to form an intact ribosome, and it requires energy from GTP, and GTP is an energy transfer molecule similar to ATP. Now, what is the difference between bacterial initiation and eukaryotic initiation? The difference between eukaryotic and prokaryotic initiation indicates that initiating amino acid is methionine in case of eukaryotes, but in case of prokaryotes the initiating amino acid is N-formyl methionine. Secondly, the initiation complex is more complicated in case of eukaryotes than the prokaryotes, because it contains 9 or more protein factors and many consisting of several units. Third difference is eukaryotic mRNA also lack in ribosome binding sequence. This ribosome binding sequence is present in the messenger RNA of prokaryotes, and that is absent in the messenger RNA of eukaryotes. So, pre-initiation complex in case of eukaryotes that binds to a protein complex, and this protein complex is associated with 5' methyl guanosine cap of messenger RNA, which scans the messenger RNA base by base to locate the AUG sequence. Let me briefly restate the main points. The process of protein synthesis that consists of 3 stages, that is initiation, elongation and termination. Initiation of protein synthesis is essentially the same in all organisms, but translation in bacteria, it begins with the formation of initiation complex, and this initiation complex, it consists of small ribosomal subunit, messenger RNA, initiator tRNA molecule charged with a chemically modified methionine that is called N-formyl methionine, and initiation factors. Initiation factors, they are involved in initiation only, but they are not part of the ribosomes. In prokaryotes, small ribosomal subunit is positioned correctly on messenger RNA due to a conserved sequence, and that conserved sequence is called ribosome-binding sequence, and this ribosome-binding sequence is complementary to ribosomal RNA present in small subunit of ribosome. Energy is provided by the GTP. When the large ribosomal subunit binds to the small ribosomal subunit, then initiation factors, they are released. Initiation in eukaryotes, that involves the formation of pre-initiation complex, and pre-initiation complex that includes the small ribosomal subunit, methionine, tRNA and initiation factors with GTP. This pre-initiation complex, it binds with a protein complex, and this protein complex is associated with 5' methyl guanosine cap of messenger RNA, and this protein complex, it scans the nucleotides base by base in messenger RNA to locate the AUG sequence. The binding of initiator methionine tRNA anticodon to the start codon of AUG triggers the release of initiation factors.